Effects of UV radiation on the growth of Brassica rapa var. Pekinensis. Industrial processes and other human activities release manufactured compounds that cause the gradual and indefinite thinning of the Earth's ozone layer. Atmospheric measurements show that the depletion of the ozone layer increased the amount of ultraviolet radiation reaching Earth's surface. This poses a major problem as it affects plants of environmental and economic importance. Napa cabbage or Pechay bagyo, Brassica rapa var. Pekinensis, will be the leaf plant sample to be utilized as it is a fast-growing pother. The objective of this study is to determine and evaluate the effects of UV radiation exposure on the growth of Brassica rapa var. Pekinensis by measuring its height, leaf size, and rate of growth within the span of a 2-week-long experiment. The aim of this scientific inquiry is to contribute information and findings on the effects of UV radiation on plant life related to ozone depletion. The plants that will be utilized in the study will locally source samples of 2-week-old soil-grown Brassica rapa var. Pekinensis. Height and leaf size of the samples will be measured prior to the start of the process. 2 pots of the samples will be exposed to UV light for 0, 30, 60, and 130 minutes every day for 14 consecutive days. The source of the UV light will be a UV box with a UV tube. All plants throughout the experiment will be grown indoors using artificial light. This study will use an experimental research design. The independent variable will be the amount of UV light exposure of Brassica rapa var. Pekinensis samples measured in minutes. The dependent variable will be the height of the plant, leaf size, and rate of growth. There will be 4 groups involved in the study. 3 groups will be experimental or treatment groups, 30, 60, and 120 minutes of exposure, and another group will be control, 0 minutes of exposure. Each treatment group will be exposed to UV radiation daily. 6 hours after exposure, the height, leaf size, and rate of growth of each group will also be measured on a daily basis. All groups will have a uniform type of soil, pot, and area. Each group will be given equal treatment and kept in the same environmental conditions as to lessen extraneous variables. After the experimentation, measurements and observations recorded throughout will be organized and tabularized for easy understanding. Findings and conclusions from the analysis of data will be communicated with teachers and supervisors of the research study.